Okay, so now let us proceed with enzymes. Okay. So now let us proceed with enzymes. Why is this not working? Okay, so now let us proceed with enzymes. So as you have learned from the previous lecture, enzymes are globular proteins, diba? So we have two types of proteins, diba? Based on structure, we have globular, and we also have the fibrous proteins. So usually fibrous proteins are those that they usually just contain secondary structure, only up to the secondary structure. Okay, now for those proteins which have tertiary or quaternary structures, they are their structure is globular. So that is why most enzymes are actually globular proteins. So here in this slide, it's showing you that enzymes are key to life, and that is true. In fact, if there are no enzymes, then there would be no life. So what is an enzyme? Again, an enzyme is a globular protein which functions as a biological catalyst, speeding up reaction rate by lowering activation energy without being affected by the reaction it catalyzes. So, first, it is a globular protein. Ang globular proteins, they have either a tertiary structure or a quaternary structure. Okay, so that's a requirement. So that is one property of an enzyme. Another thing is an enzyme is a biological catalyst. So from organic, we have platinum catalyst, we have palladium catalyst, diba? You use those inorganic molecules as catalyst. So an enzyme is still a catalyst, but it's not an inorganic catalyst. It is a biological catalyst. So it's the catalyst inside the body. It's a catalyst that is involved in biochemical reactions. Okay? So from basic chem, you know that a catalyst is a substance that speeds up a reaction. So it increases reaction rate by lowering activation energy, and it is not affected by the reaction it catalyzes. So meaning what its initial state will be retained. So mobind na siya sa substrate, and then after the reaction has finished, then mabalik ra gihapon ang identity sa enzyme. So what do enzymes do? So enzymes act as catalyst in cellular reactions. So what does a catalyst do? A catalyst will speed up a reaction by lowering the activation energy. So this broken line here, this is now the mao na ni ang path nga gi-follow if ang reaction contains a catalyst. So this is the activation energy. So if walay catalyst, mao na ang energy nga kinahanglanon ni obtain para mo proceed ang reaction. But if naay catalyst, mao na lang ni ang energy nga this is what we call as the activation energy nga kinahanglanon that has to be overcome para mo proceed ang reaction. So an enzyme increases the rate of a reaction by lowering the activation energy. Okay, so enzymes are protein in nature. Is this a statement or a question mark? Well, yes, enzymes are globular proteins, but other enzymes are actually RNA molecules. So there are RNA molecules with enzymatic activity. That means that not all enzymes are proteins. Some of them are RNA. Okay? So that's what we call as the ribozymes. And although most enzymes really are globular protein, but that means this statement is not really true. Not all enzymes are proteins, okay? Catalytic behavior of any enzyme depends upon its primary, secondary, tertiary, or quaternary structure. Well, you know this because from our previous lecture, we've said that the structure of a protein determines its function. Enzyme is a protein, so it is included in that statement. Okay, so it applies. That statement applies to enzymes. Enzymes of digestive tract and those found in blood are present in inactive form called zymogen or proenzymes, and we will see to it later what these zymogen or proenzymes are. Okay, this is proenzymes. So the property of an enzyme class is that it has a binding site. An active site, sorry. So an active site is a region on the surface of an enzyme to which substrates will bind. So if this is the structure of the enzyme, so these are composed of polypeptides nga tertiary, so managanaan na siya. Okay, so it might be like this. So if this is the structure of the enzyme, this site here is what you call as the active site. The active site is a site where is a region on this enzyme, on the enzyme molecule, where a substrate could bind. So a substrate class is just a biochem term for a reactant. So it's the reactant that could bind to the enzyme. That's why it's termed as a substrate. Okay? Enzymes are highly specific for the type of the reaction they catalyze and for their substrate. That means that no two reactions could have the same enzyme. Okay, so for example, we have that sa carbohydrates, diba? We have amylase. It's an enzyme that breaks down amylose. It could not break down lipids. It's just an enzyme that breaks down amylose. It could not break down glycogen. It's the enzyme that breaks down amylose. Okay? So, they are highly specific, meaning nagid sila specific nga substrate o specific nga reaction that they catalyze. Okay? So, enzymes with highest specificity, meaning kung unsa to ang pinaka-arte nga enzyme, like pinaka-arte nga, pinaka-pilian nga enzyme, and accuracy, are involved in copying an expression of genome. So, the enzymes involved in DNA class, in DNA replication, transcription, and translation, those enzymes are the enzymes with the highest specificity. So, of course, dapat na-adjud nga to ang pinaka, kung sa ato pa, pinaka, like, high-end or like, pinaka high-end nga enzyme. So, naana siyang it's involved in these processes, in the expression of genome, copying and expression of genome. Because imagine if cheap nga enzyme ang imong ibutang sa copying of genome, then there's a possibility that ang pag-copy sa DNA will be very erroneous. Kay, of course, you won't expect much from a cheap enzyme, di ba? Cheap, it's just a metaphor, ha? It's just a metaphor. So, what I mean by cheap is that it does not have a very high specificity and accuracy. So, in case of gene expression, then we have to use the enzyme with which is the most accurate and the most specific nga enzyme, diba? So, in fact, these enzymes have proofreading function. So we will know that when we go to nucleic acids, when we go to DNA replication, transcription, and translation. Okay, so an example of that is DNA polymerase. So some enzyme class require non-protein molecule for their activity. So enzymes are conjugated proteins known as holoenzyme. So to understand this class is that a holoenzyme is an enzyme that contains an apoenzyme and a cofactor or a coenzyme. So for example, we have hemoglobin. So we know hemoglobin has 4 subunits, diba? 2 alpha and 2 beta, and it has an iron 2 plus the middle at the center, diba? So if you just have these 4 subunits, this is what we call as the apoenzyme. So if it does not contain other things, it's called an apoenzyme. But hemoglobin needs a cofactor to function. So it needs the iron 2 plus. It's a cofactor, and so together with the iron 2 plus, this whole thing is what you call as the holoenzyme. So ang holoenzyme are conjugated proteins. So enzyme without non-protein part is inactive, and it's called an apoenzyme. So without any cofactor, that's why it's called an apoenzyme, and it's not active. It cannot function without that cofactor, because in fact a lot of enzymes need cofactor or coenzyme. So these cofactors, they could be inorganic like iron, or they could be organic. Okay, so now let's proceed to mechanisms of enzyme action. So those these things are just we're just trying to introduce this the structure and the function of the enzyme. Okay, so now let's proceed to the mechanism of the enzyme action. So the enzymatic reactions takes place by binding of the substrate with the active site of the enzyme molecule by several weak bonds. So meaning you have this enzyme, and this enzyme will bind to the substrate. So if this is the enzyme, then the substrate must bind to this active site of the enzyme, diba? So it should be able to bind like that. So this enzyme will bind to the substrate. So this formation, for example, this one is now what you call as the enzyme substrate complex. This is still the complex. You have not formed the product yet. So when the reaction finishes, the enzyme will go back to its original form. So its original form, and then it will form a product. Okay, so the substrate has somehow converted into product. Okay, now, so this is how the enzyme this is how an enzymatic reaction takes place. Now, formation of an enzyme substrate complex is the first step in the enzyme catalyzed reaction. So mao jud ni ang first step. An enzyme and substrate will bind together to form an enzyme substrate complex. That's the first step. And then this EX complex will be converted into product and the free enzyme. Okay? Now, we have different models in how the enzyme would bind, because diba ang first step is that the enzyme will bind to the substrate. Mao man ni ang first step. So sa enzyme substrate reaction, this is the first step. So there are models as to how this first step could happen. So first is the lock and key, or the template model. So this is more commonly known as the lock and key. Lock and key meaning it looks like this is a lock and this is a key. The substrate is a key and this is a lock, or whichever. Okay, so it just means that the active site is just a perfect fit for the substrate. Okay, so that's why it's called a lock and key model. So another model, a second model, is called the induced fit model. So it's a little bit more complicated than the lock and key. So in the induced fit model, the substrates will enter the active site. Okay, and then enzyme changes shape, so its active site embraces the substrate's induced fit. Okay, so for example, these two are the substrates. So these substrates will enter. So they are not yet perfect fit, but once they enter, the active site of the enzyme will change conformation such that ma-accommodate niya ang such that it can accommodate the substrates perfectly. Okay? Now, so substrates held in active site by weak interactions such as H bonds and ionic bonds. Now, after that, the active site, okay, the active site can lower the activation energy and speed up a reaction by acting as a template for substrate orientation. So stressing the substrates and stabilizing the transition state, and it provides a favorable microenvironment, so it participates directly in the catalytic reaction. So after it accommodates, diba? Initially the enzyme does not really have the exact shape. It's not like the lock and key wherein perfect fit ang enzyme ug ang substrate ng ang enzyme active site ug ang substrate. It's not like that. In this case, the substrate ug ang enzyme active site is not the perfect fit. But once ang substrate mo approach siya sa enzyme, once it's approached the active site, then this active site will change its conformation such that mo accommodate siya sa substrate. Okay? And then after that, the substrates while it's bound to the enzyme, the substrates will change conformation such that it can form products. So ma-release niya ang products, and then the active site will now be again available for new substrate molecules. Okay? So the enzyme still has its original conformation. So originally daw ay ani siya, so it's not perfect fit. They are not perfect fit, but then mo conform siya. Okay? So inig ka end after niya ma-release ang product dere, the enzyme will go back to its original shape. Okay? So again, this is a graph showing you the catalyzed and an uncatalyzed reaction. So with enzyme, if you look at the activation energy, sorry, this is the activation energy. Gikan sa reactants nga energy. So this one, if this is the activation energy with enzyme, and this is the activation energy without enzyme. So that's why nga slow ang without enzyme, because tanawa kung pila pa ang energy, it's a huge energy to conquer, di ba? Compared to a catalyzed reaction wherein gamay ra ang energy nga iyang i-conquer para mo maka-form og enzyme substrate complex. Okay, so enzymes class have nomenclature. So this organization IUBMB, they recommended system for of nomenclature for enzymes. Okay, so this is a very good system para to identify the enzyme, because there are so many enzymes that it might be difficult kung wala sila'y proper or wala sila'y systematic nga naming, di ba? So that's why there is a systematic name, but we also have trivial name or common name. Okay? So sa systematic name, ang each enzyme is characterized by a code number called enzyme code. So kada enzyme class na siya EC niya. So and it contains 4 digits separated by a dot. So example ECMNOP. But enzymes are represented kanang by numbers, like EC5315, something like that. Okay? So mo na ang this one is a specific enzyme. This is only one enzyme. We can have EC5416. Okay, this is another enzyme. So that's how enzymes are named. So ang first digit, so this one represents the class. The second digits represent the subclass, the third digit stands for the subclass or subgroup, and then the fourth digit gives the serial number of that particular enzyme. Okay, so for example, for hexokinase, this is the first step. This is the first enzyme in glycolysis. So it's 2EC2711. Okay, this is the systematic name of enzymes. And if you want to know more about enzymes class, you can just visit Brenda. Brenda, and just type this in Google, and you will see the Brenda enzymes. This is the how should you call that? This is the website for all the enzymes that exist. So it will give you the cofactor of that enzyme, the optimum temperature of that enzyme, all the details that you might need. Okay, so those are all experimental results or experimental details. Okay, so according to the IUBMB system of enzyme nomenclature, diba? Ang enzymes are grouped in 6 major classes. Okay? So that's why you have this one. Okay? So ang katong first nga digit is actually the class. So there are 6 classes of enzyme. We have oxidoreductase, there are class 1. We have transferases class 2, hydrolases class 3, lyases class 4, isomerases class 5, and ligases class 6. So we use the mnemonic OTLIL to remember these classes. So oxidoreductase, meaning they are the enzymes. This class of enzymes catalyzes redox reactions. This one catalyzes the transfer of one group to another. We have hydrolases. This class of enzymes are involved in hydrolysis reactions, and then lyase, and then isomerase, and then ligase. Okay. So next is the factors affecting reaction velocity or speed. Okay, so it means these factors affects the speed of the reaction. So first is the temperature, of course. Another thing is the pH, or the hydrogen ion concentration, diba? The hydrogen ion concentration is related to the pH by negative log of H+. So it's the hydrogen ion concentration. We have the substrate concentration. Substrate concentration meaning ang concentration sa reactant nga mobind sa enzyme. And then we have the enzyme concentration. We have the products of the reaction. So remember, the products of the reaction could also affect the reaction velocity or the reaction speed. The presence of an activator or inhibitor, allosteric effects, and time. So we will go into this one by one. Okay, let's start with temperature. Now, so the y-axis is the reaction speed and the x-axis is the temperature. So a lot of enzymes class, these things are all empirical. Empirical meaning based on experimental results. Okay, so a lot of enzymes class have different. The enzymes have different optimum temperatures, but generally, ang enzyme class, they have low activity, so low reaction speed, low reaction velocity at the start, so at lower temperatures, and then they will reach to their optimum temperature. And then if you go beyond that optimum temperature, optimum meaning they have this highest activity at that temperature. If you go beyond that optimum temperature, then the enzyme could decrease. The reason the enzyme activity decreases, the reason for this could be denaturation, because if init na kaayo, di ba, ma-denature, di ba, pH and temperature could denature, di ba? Those are two of the factors that could denature an enzyme. So because probably because of high temperature, then that could have denatured the enzyme, thus lowering the activity or the reaction velocity. Okay. For pH, so for pH class, many enzymes also could have same reaction velocity and pH. So again, ang enzymes class, so this is just a single enzyme. Okay, so some enzymes only have one optimum pH, but other enzymes like this one could have two optimum pH. Okay, so again, it's the same thing, the same reason. So could be that the enzyme here has lost its activity due to denaturation, but then we could renature an enzyme. So maybe the enzyme has been activated again once it reaches then this pH. Okay, so the thing is that pH could affect the enzyme activity. Okay, so this enzyme is pepsin. Okay, so pepsin has two optimum pH. Sorry, this is pepsin and this is trypsin. Sorry, these are two different enzymes. Okay, I'm so sorry. Now, the rate of the reaction or velocity is directly proportional to the enzyme concentration when sufficient substrate is present. Now, what this means is that if you have enough substrate, so for a given substrate, okay, same amount of substrate, okay, this is same amount of substrate. If you have the enzyme concentration is directly proportional, meaning that with the same amount of substrate, if you use small amount of enzyme for this one, but huge amount of enzyme, large amount of enzyme for this one, then this will give you a higher velocity or a higher speed, reaction speed compared to this one. Okay, so it's proportional to the enzyme concentration. So it's this one, enzyme concentration and velocity. And then accumulation of product in a reaction causes inhibition of enzyme activity. Okay, so that is why the product is also one of the factors that could affect the reaction speed, because it could inhibit the enzyme activity. So once you have too much of the product, that product could be toxic to the enzyme. That is why ma-inhibit ang activity sa enzyme. Okay? Next is the effect of substrate concentration. So this is the reaction velocity and this is the substrate concentration. So if you have higher substrate concentration, so as you can see from here, magkataas ang substrate concentration, then it could attain the maximum rate. Okay? Next, let's have the enzyme kinetics. So from the word kinetics, it's based on rate. Okay? So enzyme kinetics is the study of reaction rate and how they changes in response to change in experimental parameter, and this is what we call as kinetics. Okay, so the amount of substrate present is one of the key factor affecting the rate of reaction catalyzed by an enzyme in vitro. Okay, so if you have gamay ra imong substrate, of course, it could be good for the enzyme or it could be bad for the enzyme. Okay, so the amount of substrate is a factor on the rate of the reaction. In vitro class, it means in the lab. Okay, it's not inside an organism. If it's called in vivo, it's inside an organism. If it's in vitro, it means in the lab. Okay, and if it's in silico, meaning it's in modeling, like computer modeling. The effect of substrate concentration on reaction velocity. So as you increase the substrate concentration, so the unit is millimolar. So while nag-increase ka sa substrate concentration, nagkataas ang velocity sa enzyme until such time that you reach this maximum velocity of the enzyme. Diba? Now, take note of these terms. We have 1/2 Vmax, Km, and this initial velocity, and this substrate concentration. See, these are the 5 important terms in this graph. So, this model class involves one substrate molecule. Okay, so usara to ka substrate ng gikuan. In order to understand the kinetics, this person, Michaelis and Menten, these are two people. So Michaelis Menten, they provided kinetics or the mechanism as to how that reaction happened, how the kinetics of that reaction happened. Okay, so we have a single substrate and an enzyme. Now, once it forms the enzyme substrate complex, the rate of that reaction is proportional to this rate constant K1. Okay, so that's proportional to that rate constant. And then, for the second step, the breaking of this ES complex to form E and P, or the enzyme and the product, is proportional to this K2. Okay? Now, according to Michaelis-Menten, this reaction, the first reaction, could proceed backwards. So, diba, ang graph ane is that this is energy and this is time. So, this reaction goes there, diba? Mo proceed ang reaction. So, if you have this one, so from reactants, so mao ni ang reactants dire. And then it should form the enzyme substrate complex here and then enzyme plus product here, diba? So, if you have here this is the first reaction. This enzyme and substrate must bind together to form the enzyme substrate complex. So up to here, it should be the ES. This is the first reaction. This is a step 1, diba? It's this reaction. So according to Michaelis-Menten, once the enzyme substrate complex is formed, it could either proceed to form this E plus P. It could either proceed to form products, or it could either proceed back to form back the enzyme plus substrate. This is what Michaelis-Menten used in explaining or creating the mechanism as to how the kinetics of an enzyme substrate, an enzyme catalyzed reaction occurs. Okay, so S is the substrate, E is the enzyme. These three are called the rate constants. Okay, so from that graph class, from the graph of Michaelis-Menten, this is actually the graph. So from this, from Michaelis-Menten concept, they were able to provide this Michaelis-Menten equation. This equation class defines the quantitative relationship between the rate of an enzyme reaction and the substrate concentration. Because we've said nga the substrate concentration greatly affects the speed of the reaction or the velocity of the reaction. Diba? So this is what Michaelis-Menten is trying to explain. It's trying to explain in a quantitative manner. Okay, so mathematical ni ang Michaelis-Menten. So this is the Michaelis-Menten equation. So initial velocity VO is equal to Vmax, that's the maximum speed, times the substrate concentration over Km, which is the Michaelis-Menten constant, and that's equivalent to this one. So the Michaelis-Menten constant is a ratio of the rate constants. Okay, that's K minus 1 plus K2 over K1. And that's plus the concentration of the substrate. Okay? Now, because the Michaelis-Menten this plot is quite complicated because it's quadratic. If you can see from this plot, a quadratic is like this, diba? Is this quadratic though? Okay, so it's not linear, diba? This is not linear. So Meaning, if this is not linear, that means that it's complicated to compute. Diba? The easiest equation to compute is the linear equation. So, what Lineweaver and Burke? So, this is the Lineweaver and Burke. What they did is they converted that plot into a linear plot. It's just a linear representation. They are the same, but Lineweaver and Burke made it simple and made it convenient by making it into a linear equation. So instead of using this equation, they provided an easier equation. So this is just a linear representation so that it's more accurate and it's more convenient to determine the Vmax and the Km. Okay, so this equation is obtained by taking the reciprocal of both the side of Michaelis-Menten equation. So what they did is just they take the reciprocal of the x-axis, the y-axis and the x-axis. So 1 over s and 1 over VO. So when they plotted those values, the same values, they were able to get a linear equation. So a linear equation could give you, a linear plot could give you this linear equation, which is equal to y equals mx plus b. And now it's easier to compute for Km over Vmax, which is m, and then we have x and b and then y. It's now easier to compute for these terms. Diba? Okay. Aside from okay, so enzymes work, diba? Enzymes will catalyze the reaction, they will decrease the activation energy, they will increase the speed of the reaction. But aside from that, enzymes can also be inhibited. Inhibited meaning pugngan ba? Inhibit, okay? So there are substances that could inhibit the function of an enzyme or the activity of an enzyme. Okay, so any substance that can diminish the velocity of an enzyme catalyzed reaction. So a lot of these, like for example drugs, so these are the action of drugs. A lot of drugs class, ang function nila is to inhibit the enzyme. Kaya para dili to mo proceed ang katungan reaction which might have caused the disease. So those are the principles of making drugs. That's just some of the principles. We also have antibiotics, poisons, and antimetabolites. Okay? So it's utilizing the concept of enzyme inhibition. This is also useful in understanding the sequence of enzyme catalyzed reactions, metabolic regulation, studying the mechanism of cell toxicity produced by toxicants. So in fact, when you go to research class, there are so many things, so many experiments that you'd need to inhibit that enzyme kay para makahibalo ka kung unsa ang in order to study maybe the other reaction baron, okay, that involves the same substrate. Okay, so this is useful in research, and then it also forms the basis of drug design. Okay, so if you're interested in drug design or even drug delivery, the topic of enzymes is very useful. Okay, so we have two types of enzyme inhibition. We have reversible and irreversible inhibitors. So ang inhibitor mao na ang substance nga maka-inhibit. Okay? So ang reversible inhibitors class, we could classify them as competitive, non-competitive, or uncompetitive. So sa competitive, it's very simple. Ang competitive is that imbis na ang substrate, so this is the substrate and this is the enzyme. Instead nga ang substrate na mo-bind siya dito sa enzyme, there is this competitive inhibitor. So usually competitive inhibitors have almost the same structure as substrate, usually, because instead of ang substrate ang mo-bind sa active site sa enzyme, this competitive inhibitor will act like it's the substrate and bind to the active site and bind to the active site instead. So it's like impersonator ba? Okay, so here you have an enzyme, this is the enzyme, and you have the substrate. Okay, so instead of forming this, in the presence of an inhibitor, it could form the inhibitor-substrate complex. So the reaction will not proceed because it will inhibit. Okay, so once mo bind ang competitive inhibitor dere, the substrate could not bind anymore. Diba? Because it's already filled with a competitive inhibitor. Next is that another reversible inhibition is called the non-competitive inhibition. So non-competitive meaning they are not competing for the active site. So the substrate here, ang substrate, could have bound to the active site, diba? Supposedly, the substrate will bind to the active site. But because of the presence of a non-competitive inhibitor, this non-competitive inhibitor will bind to another site of the enzyme. So, for example, this one, mo bind siya dire, and because once it binds here, once this inhibitor binds here, it will change the conformation or the shape of the active site such that the substrate could no longer fit in the active site. So that's what you call as non-competitive, because they are not competing for the active site, but still the substrate could not bind. Next is the uncompetitive inhibition, and this is less common, but in uncompetitive inhibition, the inhibitor only binds to the enzyme substrate complex. So it does not bind to the enzyme, but it binds to the enzyme substrate complex. So it does not have the capacity to bind to the free enzyme. Okay? So because it does not have the capacity to bind to the free enzyme, so it could not be overcome just by increasing substrate concentration. Okay? An effect anemia class is that both the Km and the Vmax are reduced. So this is how uncompetitive inhibition takes place. So you have an enzyme, and then this enzyme will bind to this enzyme will bind to the substrate. So this is the substrate. So now you have formed this ES complex, enzyme substrate complex, diba? In the presence of the uncompetitive inhibitor, this uncompetitive inhibitor will bind to this enzyme substrate complex. Okay? So these are the plots. I will upload the PPT that I have used during this discussion so that you can review. Okay, so here Vmax is unchanged, Km is increased. For non-competitive, the Vmax is decreased, the Km is unchanged. But for uncompetitive, both Vmax and Km are decreased. Okay, next. An enzyme might work so much, or it might work very little, diba? Kanang hinay ra kay siya para sa ato ang usage, or paspas ra kay siya para sa atong usage. So that is why these enzymes also have to be regulated. Okay? One form of regulation is phosphorylation. So there are lots of enzymes that have to be phosphorylated para mo activate pa ang enzyme. So for example, a kinase enzyme. So kinase is an enzyme that is used para maka-add ka og ATP. That's an addition of one ATP. Mo na ang kinase ng enzyme. So a kinase activates an inactive enzyme by phosphorylation. This is just one example of regulation of enzyme activity. So in A here, in figure A, this is still an inactive enzyme. This is an enzyme, but it's still not functional. Once a kinase enzyme acts on it, so addan siya phosphate, because a kinase will add phosphate to that substrate. So the enzyme will be activated. Okay? We also have a phosphatase. So gikan sa gikan sa active enzyme, I mean inactive enzyme, so it could remove phosphate niya, ma-active pud siya. Okay, so that's what we call as the regulation of enzyme activity. This is just one example. Okay, so the rates of enzyme catalyzed reactions are controlled by regulatory enzymes that increase the reaction rate when more of a particular substance is needed. So if we need more of that product, then we have to regulate the enzyme such that iyang paspasan, di ba? So we need this regulation, di ba? We need to regulate these enzymes. Or if paspas ra pud kaayo ang pag-work sa enzyme such that it's forming too much of the product, nya dili na to kinahanglanon ng product, then we have to regulate the enzyme. We have to decrease the reaction rate. Diba? So that is why enzyme regulation is very important. Now, enzyme activity can be regulated by allosteric enzymes, feedback control, and covalent modifications. So, let's go first to allosteric enzymes. Allosteric enzymes bind with a regulator molecule at the allosteric site that is different from the active site. Okay, so for example, if you have this enzyme, and this is the active site. Okay, this is the active site of the enzyme. For example, ang allosteric enzymes, they bind with a regulator molecule. So some molecule will bind to an allosteric site. So it's not this site. This is the active site, but a regulator molecule will bind to this active site. Okay, I mean allosteric site. So an allosteric site class is a site other than the active site. Okay, that's what we call as an allosteric site. So what happens is that once that regulator molecule binds to this allosteric site, then it could change the shape of the enzyme, and so it will also change the conformation of this active site. Okay, and it could be a positive regulator, meaning that if it's a positive regulator, it could change the shape of the active site such that mo favor siya nga mas ma-active ang enzyme, or it could be a negative regulator such that ma-change ang shape sa active site para dili siya mo activate og maayo, so to lessen the activity. Okay, so it's actually, if you can see, it's a little bit like a non-competitive inhibitor, but the difference is that it's just a regulator molecule. It's not a competitive substrate. It's just a regulator molecule ang mo-bind sa allosteric site. Okay, so it's this one. This is for positive allosteric regulation. So this is for positive. Okay, this is the active site, and this is the allosteric site. So this is the effect, the regulator molecule. This regulator molecule binds to this allosteric site, and once it is bound to this allosteric site, then the substrate could fit perfectly. So before, ingon ani ang shape sa substrate, it could not fit perfectly, and then once it is bound to, once this effector this regulator molecule has bound to the allosteric site, then masud na og ma-perfect fit na ang substrate. Okay, so it's called positive allosteric regulation. We have a negative allosteric regulation wherein if once ang regulator molecule binds to this allosteric site, once it binds to the allosteric site, it changes the shape of this of this active site. So from dako it changes to the shape of this active site such that maglisod na og sulod ang substrate, so it won't fit anymore. So it's a negative allosteric regulation. Okay, so ang positive allosteric regulation, we need that if we want to produce more, if we want to activate the enzyme. For negative allosteric regulation, if we want to partially deactivate the enzyme. And next is feedback control. So in feedback control, when the end product is high, the end product of a series of reactions acts as a negative regulator and binds to the allosteric site. So this is what we call as a feedback control. Okay, so the substrate cannot bind to the active site, and production of all the intermediate compounds in the subsequent reaction sequence stops. So for example, you have this reactant 1, and then it's formed, there is an enzyme 1 here, so there's enzyme 1, so this is reactant 2, and then it will form product, and then we have enzyme 2. Okay, sorry, this will form product, diba? So ang feedback control class is that when the end product level is high. So for example, daghan na kayo ang end product, so daghan na ni, and the end product of a series of reactions acts as a negative regulator and binds to the allosteric site. So, because daghan naman ni siya, this will act as a negative regulator, meaning it could bind to the allosteric site of this enzyme. Okay? So, it will bind to the allosteric site of this enzyme, so mahunong ang reaction. Okay? This is what you call as feedback control. So, okay, dara. Feedback control when the level of end product is low. Okay, this is another example. Kung end product is low, katong ganiya high to. When the end product is low, the regulator dissociates from the allosteric site on the enzyme, unblocking the active site. So, here, di ba ni bind man to ang regulator. Okay, so let's follow this. Okay, so we have this enzyme, and then this is the substrate. So the enzyme and then the substrate binds. This is the allosteric site. Okay, and then let's start with this one. I think it starts here. Okay, so this is the substrate, and then we have an enzyme. Okay, this is the enzyme, and then we have the substrate. So supposedly the substrate will bind here, but the substrate could not bind because it has formed this negative regulator. Okay, so na-form ni ang negative regulator. So we have enzyme, diba? And then mo bind ang substrate here. This is the active site. Ni bind ang substrate sa active site, and then ni-form siya intermediate, and then after that so ang intermediate will bind here. So these are just series of reactions. Okay, so this is reaction, reaction, reaction. Okay, these are series of reactions, and then after that ni-form na po siya ginani, and then it formed this negative regulator. So due to the formation of this, ang end product kay it's a perfect fit to this allosteric site, di ba? So it will bind to the allosteric site of the enzyme. So it changes the active site, so the reaction stops because of the presence of because of the binding of this allosteric negative nga regulator. So na change ang active site sa enzyme. Okay, so this is what you call as feedback control. So ang feedback control class, ang difference ra niya in allosteric regulation is that in feedback control, it's a product. It's due to the series of reactions, and it's a product of that series of reactions that will regulate. Okay, that's why it's called feedback. Kay gikan ra pod sa iyang own nga series reaction, di ba? So gikan ra sa iyang binuhatan, own niya nga binuhatan. That's why nahunong ang reaction. Lastly, we have covalent modification. So it's another way in which enzymes are modified. So from the word itself, it's modify, okay, and modified by covalent bonds. So it's the enzyme activity is modified by covalent bonds to a group on the polypeptide chain that are formed or broken. So this modification is reversible. So for example, zymogens or proenzymes, katong ganiha, like the insulin. So zymogens or proenzymes are produced in their inactive form. Okay. So inanin siya class. Example tani kay digestive enzymes and proteases. Okay, so what happens actually is that once a zymogen is formed, it's transported to where the active form is needed. So dili sa siya active ang i-produce. Okay? So let's just have this example. So for example, ang hormone nga insulin. So di ba we've had insulin in carbohydrate lecture. So, di ba, i-synthesize siya sa pancreas once taas kaayo ang blood glucose level na to. Di ba? So, ang pancreas mo, secrete siya og insulin. So, actually, ang katong gisekrete nga insulin sa pancreas is a zymogen. It's a zymogen and it's specifically called a proinsulin. Okay? That is not yet an active insulin. So, it only becomes biologically active when the polypeptide chain of 33 amino acids is removed by peptidases. So kanang ang katong proinsulin nga gi-secrete sa pancreas. So dili to active pa ha, ika-secrete sa pancreas, it's not yet active. So actually natoy modification. So this is the inactive form of the insulin. So the insulin contains a polypeptide chain, this yellow chain connecting A and B peptide chains of insulin. So this is the A chain. So, mao ni ang A chain and this is the B chain. Okay? So, para ma-activate ni siya class, it has to be the polypeptide chain must be removed. So, mao ni ang covalent modification. So, this is the modified. Mao ni ang covalent modification nga gibuhat. So, by doing this, it's inactivating the insulin, di ba? It's called proinsulin. This is the inactivated. Ang general term niya is a zymogen. Okay, so para ma-activate na siya, dapat i-break sa niya ang covalent bond. Okay? Another thing is phosphorylation, which we have discussed in the first slide, diba? So, inactive pa na siya ng enzyme. This one, inactive pa ni siya ng enzyme, but once i-phosphorylate siya, then it becomes an active enzyme. So, this is still called a zymogen. And then it will turn into an active enzyme. Okay? Okay, so you indicate whether the following statements describe an enzyme regulation by an allosteric enzyme, a zymogen, or covalent modification. So, in an end product attaches to the regulatory site of the first enzyme in the reaction sequence. So, this is what you call as an allosteric enzyme by feedback control, di ba? Letter B, it's proinsulin forms in the pancreas. Well, we've had that as an example. It's a zymogen. Okay? And then phosphorylase kinase deactivates pyruvate dehydrogenase. It's called covalent modification.